Okay, so this is the last part of chapter 5. This is part 4. And in this part of the chapter, we will focus on bulk transport. Okay, and so bulk transport is going to be the movement of large molecules or large amounts of molecules or particles. So it's moving a bulk amount of material across the membrane. It could even just be water, or it could be like a whole bacterial cell, or a bunch of sodium ions all at once. Okay, so it's the movement of a large volume of molecules or particles all at one time. This, like active transport, requires energy. In fact, it is a form of active transport. So there are two main types. There is endocytosis and there is exocytosis. And so we're going to start with endocytosis first. So when you hear the term endocytosis, I want you to think internalizing. And so endocytosis is when we move stuff, whatever that stuff is, into the cell in bulk. Okay? When this happens, there's kind of a series of events. And so what's going to happen is the plasma membrane at that site is going to, what we say, invaginate, which means to kind of fold in, forming a pocket. And that pocket's going to form around whatever's being brought in, whatever that target particle or cell or fluid is. So it folds in, forms a pocket, and then what happens is that pocket pinches off the membrane, and we end up with a vesicle. Okay, so a membrane-bound vesicle that's been formed from the plasma membrane and is carrying the contents from outside that cell. Okay, and I'll show you a picture of that in just a second. There are three kind of categories or three types of endocytosis. We've got phagocytosis first, pinocytosis, and then lastly, receptor-mediated endocytosis. And so we're going to go through all three, and I've got a figure for all three, but keep this in your mind. When you hear phagocytosis, I want you to think cellular eating, so bringing in large particulate stuff. Pinocytosis, I want you to think of as cellular drinking, so bringing in a large amount of fluid all at once. And lastly, receptor-mediated, it's phagocytosis, so you're bringing in a large volume of stuff or a large thing, whatever that is, but it's in a specific way. And so before anything is brought in, that stuff has to first bind to a surface receptor. And so we say that molecular recognition is involved. So whatever's coming in has to first bind to a receptor. So it's very specific, and then the process begins where the membrane folds in and so on and so forth. So again, let me show you pictures of these three types. And so again, the first is phagocytosis. And so what do I mean by cellular eating? So again, this is the cell bringing in a large particle, or maybe even a whole bacterial cell, or again, a large amount of particles. So this is particulate matter. That is coming in, not fluid, which is pinocytosis. And so this represents the plasma membrane here in yellow. And this lightish yellow beige is the cytoplasm. And then outside of the cell here, represented in kind of this blue, is going to be the extracellular fluid. Okay? And so what we're bringing in, it looks like maybe a bacterial cell. They're just calling it a large particle. And so it's near the surface of the cell. It doesn't need to bind to anything, so it's not specific. But this membrane is going to, again, invaginate, kind of fold in and form eventually a pocket. And sometimes the membrane, you can kind of see that it's jutting out a little bit. We call these pseudopodia. Sometimes the membrane will kind of grow out and kind of wrap itself around whatever's being brought in. Pseudopodia means false feet. They're the same structures that would be used in cellular movement. And items of the cytoskeleton, remember we talked about that in chapter 4. So in this case, it would be intermediate filaments. They're going to wrap themselves around whatever's being brought into the cell. Okay. And so what we end up with in the end, so again, this invaginates, and then it's eventually going to pinch off of the membrane, and we end up with a vacuole. And the membrane seals itself right up, okay, afterwards. And so this vacuole carrying its contents then enters the cytoplasm. And usually it will fuse with the lysosome. And the lysosome, remember, contains digestive enzymes, and then those enzymes will kind of chew up whatever that particle is that was brought in. Okay, so the second type of endocytosis, so again, bringing stuff into the cell, is penocytosis. And so again, when you think of penocytosis, I want you to think cellular drinking. And so this is bringing in kind of large amounts of fluid. And so, when, for example, water is needed inside the cell, and we don't have time to wait for osmosis to occur through aquaporins, sometimes water will be brought in this way. And so the same idea. So you've got your plasma membrane here. It starts to invaginate, which just means to fold in. Water molecules will come in. Maybe there's some particulate matter. And then we end up pinching the membrane, and the vesicle is now in the cytoplasm. And again, this vesicle might fuse with something else inside of the cell, or the contents inside the vesicle may just diffuse out. Okay, it depends. Lastly, our last type of endocytosis is receptor-mediated endocytosis. So remember again, phagocytosis and penocytosis, they're not specific, where this is specific. So whatever is going to be brought in has to first bind to a membrane receptor. And those receptors are going to be specific. They don't just bind to anything. Lock and key. Okay? And so in this case, the stars fit the receptor. And so they're going to bind to these membrane receptors. That's going to trigger the membrane to, again, invaginate and eventually pinch off. And you end up with our vesicle. Okay? So again, the receptor is shown here in purple. These are called clathrin molecules. So this is called a clathrin-coated pit. And then this becomes a clathrin-coated vesicle. It's just a protein involved in this process. And again, same thing. This could then fuse with something else inside the cell. It could fuse with the lysosome. Or again, the contents might just be kind of dumped out into the cytoplasm. Okay? And lastly, we've got exocytosis. And so this is the reverse of what we've been talking about. So endocytosis meant to bring in material. Exocytosis is moving materials out of the cell, again, in bulk. And so what types of things would be moved in this kind of a way? Waste products. This is a fast way to get rid of waste products. Other things that leave are secretions. And so hormone molecules that are made inside the cell will be secreted this way, as well as neurotransmitters if we're talking about neurons. Okay, so what happens here? Vesicles carrying whatever this is, waste products, or if it's hormones or neurotransmitters, these vesicles are probably coming from the rough endoplasmic reticulum. They're going to travel through the cytoplasm, right, traveling on elements of the cytoskeleton with the use of motor proteins, all those things we talked about in chapter 4. And eventually these vesicles will make it to the membrane. When they get to the membrane, they fuse. Okay, so the plasma membrane and the vesicle membrane become one, and the contents inside the vesicle will literally get dumped out. Okay, and then those go to wherever they need to go. And that is it for chapter 5.